All right, so the last thing we need to talk about are the reactions of polypeptides. There's going to be two main ones that we talk about: hydrolysis of polypeptides into the individual amino acids, and then denaturation, which is not actually a chemical reaction in the sense that you're not making and breaking chemical bonds, but you are destabilizing those intramolecular interactions that we talked about that hold proteins together. So first, let's talk about hydrolysis. So this is breaking an polypeptide chain rather into its individual amino acid components. This can be catalyzed by acid, and that's the most common one, and that's what happens in your stomach. You know, we all know that we have stomach acid, right? So what's that doing for us? It's taking the proteins in the food that we eat and hydrolyzing them into their individual amino acid components so that our body can use those amino acids, you know, whether to convert them into new proteins that our body wants to make or just to convert them into energy. So this is literally breaking these covalent peptide bonds, right? So we talked about how these different amino acids are held together. So these are three different amino acids held together by peptide bonds. What hydrolysis does is cleave those peptide bonds back into their original components. So now I have, let's just color code here, this, what was formerly my what was formerly my peptide bonds carbonyl carbon, going back to being its original carboxylate carbonyl carbon. Likewise, my formerly peptide nitrogen goes back to being that ammonium ion of the neighboring amino acid. Okay, so these are the hydrolysis of the peptide bond to get back to what we started with, which was our carboxylate group and our ammonium group on those two amino acids. All right, the one that we're going to spend a little bit more time talking about is denaturation, and that's because there's a few ways to denature proteins. Okay, and by denaturation, what we really mean, I think that this picture was actually great, means we're taking the active protein that has its complicated tertiary structure where everything's folded onto itself in this really precise manner, and you have these R groups interacting with one another, and we're going to completely disturb all those interactions, right? So we're essentially going to like stretch this thing back out or make it form a unstructured kind of loopish thing going on. And there's a few ways to do that, okay? Increasing the temperature will disrupt these intermolecular interactions. Turns out that proteins are very fragile. We talked about all those complicated and interactions holding them together. Each interaction by itself is relatively weak. So it's the sum total of having a ton of them there that's really holding that protein together. So any disruption to those ability of those amino acid side chains to interact, the whole thing just kind of falls apart. So increasing temperature will do that. Most proteins have a very narrow window of temperature that they can operate at, and that's why your body goes through such great lengths to regulate its temperature. pH is another way that we can denature a protein. So your body also goes through great lengths to keep the pH of a particular cell where it needs to be. And certain cells may be at different pHs. For example, your lungs are at a more acidic pH than most of the rest of your body, but all of the enzymes in your lungs are tuned to be at that right pH, right? So if we take an enzyme out of the pH environment it's supposed to be in and put it in the new one, it will denature. Also, organic compounds or heavy metal ions, introducing those, they can disrupt the intermolecular interactions that hold together the tertiary structure. And then mechanical agitation, so just like shaking a protein really hard, can also disrupt it. And what happens when a protein is disrupted? Well, the tertiary structure unfolds, right? We talked about how proteins have this specific folding pattern that's holding them together, and that's unique to their function. You're disrupting all of that, and you're what we say unfolding the protein. By doing so, you're making it so it's no longer biologically active. It can't do its job anymore. Okay. Proteins are denatured when they're heated above 50 degrees Celsius. Sure, that's probably a good rule of thumb. Each protein is going to be unique. Each protein has its own sort of like thermal tolerance. If you are an organism that that's cold-blooded and lives in or whatever, if you're an organism that's evolved to live in a cold environment, then your temperature range is going to be different than an organism that's evolved to live in a warm environment. Again, for most of us mammals that are warm-blooded, our bodies are going way out of the way to keep a certain temperature range so that it avoids any sort of a problem. If you're running a really high fever, one of the big dangers is that you're going to overheat, and the proteins in your brain and in your cells are literally going to start falling apart because of that, okay? When we cook food, we don't actually change what you're seeing a lot of the times when you're cooking food, you're looking for the denaturation of proteins. It doesn't really change the nutritional value of it. It will make it more digestible. And then also importantly, it will kill any bacteria that's in that food by virtue of heating it up and denaturing all the proteins within that bacteria. In that same way, we can use it to disinfect surgical equipment. There's a lot of sort of things that we use high temperatures as a disinfectant for. Basically, by cranking up the heat, we can make sure that we're going to kill any organism, and how we kill it is by denaturing all the proteins in that organism. Acids and bases. So what pH does is it messes up with the hydrogen bonding and the salt bridges within a protein, right? So we talked about how one of these acidic R chains has this sort of a form, where a basic R chain has this sort of a form, and those form salt bridges. Well, what happens, so this is at pH equal to about 7. What happens when I crank the pH up to something like 14, really basic conditions? What that's going to do is remove a hydrogen from that basic protein. So it's no longer charged. Whoop, not 3, 2. Likewise, if I go and drop the pH to something really low, that's going to change my carboxylate to a carboxylic acid, and again, disrupt that salt bridge, right? So at this physiological pH, both of these are charged. However, if I increase or decrease the pH, I've broken that salt bridge because I've removed the charge from one of those two species, okay? So in general, pH will disrupt the salt bridges and hydrogen bonding of proteins, and that's why they will begin to denature. Organic compounds such as ethanol, isopropyl alcohol, so alcohols in general will disrupt the tertiary structure of a protein. The bottom line is proteins are evolved to exist in water. Water has a very specific kind of like hydrogen bonding pattern. Ethanol can form hydrogen bonds as well, but they're not quite the same. They're not the same strength. They're not the same orientation. And again, proteins are really fragile. So if you disrupt their hydrogen bonds, the whole thing just kind of falls apart. So ethanol as a disinfectant, we all know that we use these ethanol or alcohol-based gels as ways of removing any sort of virus or bacteria from our hands. What that's doing is denaturing the proteins in those bacteria and viruses by disrupting their hydrogen bond patterns. Similarly, we can use heavy metals, although these tend to be kind of bad for you. So this is a much more rare way of denaturing proteins. But what heavy metals do is they play the role of oxidizing the disulfide bonds in proteins. right? So we have these important structural elements, the disulfide bond, a covalent bond between two cysteine residues. Silver, for example, or lead or mercury, those ions will have a tendency to oxidize those bonds and break them into their two individual components. So this all of a sudden gets broken up into, and it's no longer covalently held together. And then last one is agitation. This is kind of a unique one, but also relevant to cooking. If you take egg whites, for example, and egg whites are like 100% protein. If you take egg whites and whip them really strongly, you'll end up forming actually what's a meringue. But yeah, you denature the proteins in those eggs through that whipping action. So a lot of like mechanical agitation can also denature proteins. Okay, so this is just kind of a little bit of a summary of what we just discussed here. So this is kind of your cheat sheet for protein denaturation. Do our last little learning check here. We have tannic acid, which is used to form a scab on a burn. And an egg is hard boiled by placing it in boiling water. What is similar about these two events? Well, adding an acid would change the pH of that protein, right? And what does changes in pH do? Those tend to denature a protein. Similarly, placing an egg in boiling water by boiling something or bringing it up to a really high temperature, I'm also denaturing the proteins. So denaturing proteins is what's common in these two events.